talk about neoantigens, you are really referring to a mutation that takes place in the genome of the cancer cells. You would not find such mutations in healthy cells. So if you want to develop a neoantigen-based cancer vaccine, the first step is really to identify those mutations. To identify neoantigens, to identify mutations, you need to compare the genome of a tumor cell to the genome of a healthy cell. Once you have identified the mutation, you will have a very big number of neoantigens. Potentially, they're all candidates for a vaccine, which means possible ingredients to be put inside the therapy. But for practical reasons, you need to select a subset because it's not possible to include them all in a single therapy. So the step that goes from the big set of neoantigens to an optimal subset of very few elements require you to predict and infer what's going on inside and outside the tumor cell. So the core idea is that inside our body cell, there is a machinery that essentially converts the genetic information into proteins. And the proteins are chopped into little pieces. And they eventually can be presented on the surface of the cells. Once these little chops of proteins are presented on the surface of the cell, they act as markers. They act as features that somehow are informative of what's going on inside the cell. So once we have our big set of mutations, we need to find the optimal subset, which is actually the set of mutations that have the highest likelihood to be presented on the surface of the tumor cell. And once we identify the mutations which have the highest probability to be presented on the tumor cell, well, those are good candidates to be put inside a vaccine, because we can teach the patient's immune system to search for those mutations specifically. The process that starts from the big set of neoantigen to the small set of optimal vaccine elements require you to operate a prediction of the probability of presentation. And for doing that, we use machine learning. This is really the interface in which machine learning becomes important, because we use machine learning models to predict the probability of certain neoepitopes, or little chops of proteins, to be presented on the surface of the cells.
Orchestrating a Brighter World, NEC.